I'm Richard Acton from the Abraham Reproducibility Journal Club, and as my intro said, my background is in biology, and I've worked as a postdoc in a bioinformatics core facility, and currently work as the data outputs manager for a consortium called the Human Developmental Biology Initiative. So perhaps slightly unusually, I'm more a research technical professional running a journal club than someone who's directly on the research side at present. The motivation for starting the journal club up was kind of a bunch of the practices that I was thinking a lot more about in terms of open science and data sharing and so on that came about in my work creating materials for people to use as the data outputs manager for this consortium, and translate that into sort of applied practice for people to be aware of some of these often relatively minor points about how to report data and protocols and code well, so that the reproducibility of the work can be improved. We started the journal club back in 2023, and we've been open to kind of any remote participants since about October of 2024. So if you do want to come along, hit me up and I'll send you a meeting link. So our format's a little unusual. We do 2 sessions on every manuscript that we cover. We do one where we talk about the reproducibility best practices and one where we talk about data visualization and figure design. And we do one session every 2 weeks, so it takes us about a month to actually get through a paper, which I think gives people a bit more time to kind of dip in and out of participating in it to some degree. They have time to actually read the work, and depending on how engaged they are with the journal club, they can, come to either or both sessions. The other thing that we do, which is relevant for the peer review aspect of this talk, is we try to write a blog post up about those manuscripts. It's a bit of a variable time to release, depending on how long it's taken us to actually get around to turning our conversation into a blog post. I think our most recent one is quite a while ago now, but we have a backlog of at least 3 manuscripts that we'll probably have blog posts out on at some point in the near future. And we tend to take breaks over the summer and winter holiday seasons. So that's kind of the general way it's organized. The sorts of manuscripts we tend to select are somewhat dictated by where I'm physically located at the Abraham Institute. So it's a lot of biologists of varying different stripes. So it's usually a paper from one of the disciplines that has the attention of the Institute. And whomever leads the session picks a paper. I've been disproportionate in picking papers because I don't always get volunteers. So some of it reflects my interests. And we aim to go with preprints rather than published manuscripts wherever possible, so that if we do have any feedback to offer about those preprints, it's possible maybe they will get incorporated into those manuscripts before they actually get submitted for publication, or at least get final revisions. So the other aspect of the blog is that we tend to try and use the papers and the specific things that we find in them as sort of jumping off points to talk about broader things. So not every time a blog post will be necessarily a specific critique of a given paper. It may involve that. It may involve a very minor critique of a given paper, but which we then sort of use as a vehicle to discuss some wider systemic issue and suggest practices that various other institutions might adopt to try and encourage improvements on this in those blog posts. So the sorts of things we tend to talk about when we're tackling the reproducibility of a given paper, we're tending to think of it in a relatively narrow sense of technically reproducible. If I could pick this up, what information do I need to go about attempting to reproduce this work? We don't have the resources to actually reproduce it, so it's a little bit theoretical. It's like, do we think we could reproduce this, and what sorts of information will we need? We start out with a kind of more conventional journal club discussion of, we talk about the content of the paper, so we have that context, and then we go through and look at the methodological detail and say, is there actually enough information here for us to, reproduce this bench work, as it tends to be biology? And we have data deposition. Have they deposited the data in the right sorts of places? And the other element is a code deposition. Have they put any data analysis code somewhere that's appropriate to use, and would it be able to rerun it if we found it? A higher standard, the rerunning a bit. And occasionally, where we have the appropriate expertise, I've roped our local statistician into helping him out once or twice. We'll talk about the experimental design and statistics of a paper. And this allows us to tackle a pretty wide variety of papers. Because we're doing a relatively general critique. And a few other points on the methodological detail. Yes, that's very much focusing on, like, do they actually have protocol level methodological detail? Is there a sort of step-by-step available somewhere? Like, quite frequently you'll find method sections where in the abstract there's kind of enough information, but there's a lot of missing specifics. So the bench people who come along will help us critique that a little bit. And one of the things that is a bit of a pet peeve of mine is I tend to flag issues of any aspects of the process that are in some way proprietary or closed. So you've got things like commercial media with undefined components, use of antibodies, the specific structures of which are proprietary, or use of closed source software, all of which are kind of contributing factors to potential lack of reproducibility if any of those things are no longer available, and they're also lack of inspectability, as it were. So you can't go in and query exactly what the components are and thus come up with any theoretical or logical problems that they might pose. So getting rid of black box elements. When it comes to data deposition, we don't like to see things like data available on request. It's better than it not being available on request, but it is not particularly specific. It doesn't indicate that it's archived anywhere properly. It doesn't suggest there's any kind of process where we can apply to get access to the data with some kind of data access committee. So if there are justified reasons for placing restriction, privacy considerations, and so on, then they should be included. And those are the policies for those kinds of things in journals for the most part. And yet there's an enforcement issue, as was mentioned in the previous talk. Many of these policies exist, but are enforced sparsely and poorly by the journals and by reviewers. So yes, that's if there's a data set, does it have unique, persistent, and resolvable identifiers? Is it something where you'll actually be able to get that data back at some point in the future? Just a URL is not necessarily good enough, because URLs can change and websites can go down. Code deposition, I have a whole kind of separate side project on code deposition. You can read rsspdc.org, where I'm working on checklists for this. But the sorts of questions we look at are, if you have any kind of computation in your paper where you've written some custom analysis code, which is extremely common nowadays, have you included that code? This is a part of your methods, and not including it is like leaving out an important step of your methods. And then various things for the quality of that code. Do you have it under version control? Is it on a Git repository somewhere? Can I cite it as a separate entity? Can I refer to it as its own unique identifier? Is it documented well enough that I could actually use it again or run it again to reproduce your results, even if it's not a general tool, but just enough to reproduce narrowly what you did on a system that is not your particular computer? And do you have the environment well specified? Those sorts of things. The no hard-coded absolute path one is the most common problem for re-executability of code. It's just someone's username is in the path, and it's not actually just relative to the project directory. If you fix that with an automated tool, your rate of re-execution of R scripts in a bunch of corpuses goes way up. So really simple fix, but not that commonly adopted. And so the other sort of relatively unique element of our journal club is we have a focus on data visualization and figure design. So that's what the second session is for. The first one is on reproducibility, and the second one is attempting to revisualize or rework some of the figures. This is often a quite convenient illustrative example of lack of sharing of processed data, because whilst, especially in biology, it's relatively common for certain types of data, particularly sequencing data, for people to deposit those data sets in a way that's accessible. They are not particularly good at depositing secondary data or processed data that is sort of the raw information that you need to replot your data sets. So the way in which you actually attempt to change the way the data is visualized, it's tricky. You can't just sort of get in there and replot stuff and apply some statistics and stuff that may be downstream of any issues in the upstream processing. So it's nice to have usually a pretty simple Excel spreadsheet somewhere which has the raw data that you're plotting, but not very common. So we tend to sort of simulate and make up some data that we can use to accomplish this task if the data is not actually available. So an example of the sort of thing that we might tackle is we'll look at a figure in a paper. So on the right here with a few red boxes on it is the original. On the left is the one that we tweaked for legacy reasons, where things used to be on paper and color print used to be scarce, and you'd get like one page of color printing in your journal. So you crammed all your figures into a limited space and stuck them all on one page, and it means the actual total number of figures that you have is usually far less than the semantic number of figures you have, or the sort of logical separation of figures. So you almost always in biological papers end up with these massive figures with too much stuff crammed into them. So my workaround for this, imperfect though it is wherever you can get away with it for journal formatting policies, is do some subtitles that actually split up your figure into its semantically sensible figures, where ideally you'd have them separately. This is usually around about the sort of paragraph of text that refers to the particular sub-panels of your multi-panel figure. So, descriptive title of what the thing is, and then, logically organizing the contents of the figure. And in this example, these were grouped in an order that didn't make a lot of sense for these things. The titles for the actual plots are frequently missing, and then you have to go and find them somewhere in the figure legend, which is sometimes on a different page, and the title of the individual panel is not. There's a bunch of stuff that just makes these things actively hostile to read. So there's basic visual steps you can take to make this information more digestible to the reader. And another thing that we occasionally talk about is semantic color palette design, and thinking about how you use your colors consistently in papers, because it's a useful aspect of visual communication, but people often don't pay a lot of attention to it. This is a relatively complex color palette. This is the original one, and it's representing a bunch of different things here. This is a C. elegans experiment with an Alzheimer's model with a couple of different mutants that affect it. So the sort of approach that was taken to designing this palette is very much a flat qualitative palette. So each different color represents a different group, and there's no semantics to what the color is. But there's quite a lot of information that you can encode into the colors if you're thinking about how to communicate this to your reader. So you've got a couple of aspects to this palette. So there's a sort of control and experimental condition part. So you've got your tau proteins, the aggregates in the Alzheimer's sign, that's more on the experimental side. And then you've got your control conditions, your wild type. So you can have a difference in the colors to represent that. And you've got these two different mutants. For that, you need a sort of two classes of colors that are nicely distinguishable. And then you've got sort of background information, another kind of control information that you might want to communicate. So there's a bunch of combinations of these that exist in the plot. So actually designing a palette that communicates that can be quite tricky. Generally speaking, using grays and lighter colors for control conditions works quite well. And then bolder colors for conditions where you're actually the more relevant conditions that you're sort of testing. So this is the sort of redesigned version of the figure. This is the one with the original color palette, and this is the one with the more semantically designed color palette. And in this version, you can see that the purple colors correspond to this UNG1 genotype. The green colors correspond to the NTH1 genotype. And as you're going from a bolder color to a lighter color, you're ending up with the control conditions that are of less interest. In the comparison here, these are kind of background information. The key points, for example, in this top right panel that you want to be able to draw attention to are, this is the orange one here, the tau aggregate. This is the disease state, as it were. The bold green one, that's the rescue of that state, and then the N2 is the sort of wild type control. So they're illustrating very nicely that the points that your attention is drawn to are the ones that are most relevant for this comparison, and that communicate to you what the actual meaning of those encodings is. And other points here include things like, in the original, these background strains have very non-intuitive namings. So swapping them out for a name like these tau and aggregating tau variants are much more effective at visual communication to the end reader. So that's the sort of feedback that we tend to provide on data visualizations. So the next aspect of it is we tend to reach out to authors once we've provided some kind of thoughts and put up a blog post on this. Initially we had very positive comments from authors, but we have had some pushback on a couple of instances. So I think I'm going to be shifting to a policy of sharing the blog post with the authors before we put them up on the blog, so they have a bit more of a sort of right to reply, and that we could incorporate some feedback in there to make sure there aren't any miscommunications with the authors on this point. But broadly speaking, it's been very well received. The couple of comments we got from authors is, this is a very interesting concept for a journal club, and I appreciate all the thoughtful feedback on our paper. We took your feedback into consideration and have updated several of our figures accordingly. So that was our commentary on a preprint. I haven't double checked in a little while if that's actually out yet, but it may have been published. We'll see. And then, wow, I'm impressed. Thank you for covering our paper. And also thank you for pointing out all those caveats. Of course, all of them are very appropriate. I wish I'd had the opportunity to discuss the paper with you before submitting it. I will take these comments into consideration for the future. So pretty broadly positive. We had one author kind of push back a little bit. I think part of that was a problem of communication on my end, in that the point that I was trying to make in that blog post was critical of a very narrow aspect of the paper. So this is a paper I really like. I like the conclusions of it a lot. It's been one that has informed some of my thinking about some important issues. But there was a couple of things where the sort of data sets used had some provenance issues, and I was using them to talk about systemic issues with reporting and clarity of access to data, and all these sorts of problems of fair data structure. I didn't really set the post in the context of that, so I think that that may have failure on my part may have been why the author responded perhaps a little bit less positively to that particular critique. But we had a productive back and forth, and I updated a couple of things to provide that context, and it is I think we're now on good terms. But that made me think a little bit about how to best manage this point when it's when you're commenting publicly on the work of other authors. And beyond that, I also began thinking about, okay, if we actually, it's not really the focus of what we're doing, we're not kind of engaging in a data sleuth type activity, but we may encounter error, fraud, or misconduct of some kind in the papers that we're covering. So I've given a little bit of thought to sort of how to handle that. There's you can potentially comment anonymously on PubPeer if there's a problem for some reason with commenting publicly. There's the option of reporting it to the publisher or the journal if you discover them. But I think one of the things I'd be quite inclined to do if I encountered a particularly bad case of this is try and reach out to some of the people who have experience of dealing with tackling people with this kind of thing. So if you're actually kind of setting up a larger independent exercise to run a sort of journal club blog where you're doing data sleuthing stuff, then I think it's probably a good idea to either consult with your institutional body about the legal processes there. So what legal protection do you need? Like if you're an independent media outlet, then media liability insurance is something you need if you're potentially going to be making claims against litigious institutions or people. That's probably the most risky category are the sort of independently wealthy individuals, especially ones in either your own or in friendly jurisdictions when it comes to libel laws, as it were. So making sure your claims are very buttoned down is obviously the first step in that kind of thing. But it's worth considering these factors before you publish, if you're in that space. One of the other things that we do in the blog is we're attempting to use the blog as a sort of a mechanism to learn and teach about reproducible computational best practices. So our technology stack is all open source, And the services we use, we aim to pick ones that it is, at least in theory, possible to host yourself. So we don't use GitHub, for example. We use GitLab because you can host a GitLab instance, but you can't host a GitHub instance. We have various tools that we use to collaborate for authoring of articles. Git, for example, we're using literate programming. Quarto is the tool we use there for authoring. These slides are written in Quarto, and our blog is also written in Quarto. And then we have a mechanism whereby I can automatically generate the website by pushing a Git post to the automation on GitLab, and it serves it as a web page. And then the compute environment, which evaluates any of the code that's in the blog, is used. Nowadays, there's a Nix environment that it runs, as I've been moving more to Nix because it has some nice theoretical advantages over a Docker and other technologies for computational reproducibility. For the more technically inclined, I'm happy to go into more detail there. But I tend to think of this approach to publishing as somewhat analogous to the octopus approach, actually, in that it's somewhat granular, somewhat more granular. Like software publishing is doing kind of the same sort of thing, right? There's a continuous process of the integration of review for new additions, but because software is itself always a continuously modifying artifact, it's never had the problem of having to move away from a sort of single point publication. So a lot of the processes that I think we can learn from are present already in how open source software development works. So I think adopting even much of the tooling that software developers use for collaboration on the production of articles and the review of contributions and updates to articles and so on is a fertile ground for innovation in the academic publishing space. And it has the advantage of making computational results much more reproducible if you take the right steps to designing that approach. So that's it for me on the talk side of things. I posted the slides in the Slack. There's a few more resources linked on this one. But yes, I'll take questions now. Thank you very much.